This presentation will consist of a brief introduction to the ReactIR technology, in other words, how it works and why you'd want to use it. Then we will go through several case studies taken from the recent literature that have utilized ReactIR in combination with other analytical methods to further process development or provide better reaction understanding. The first case study involves formation of an activated Lactol species. The second, the stability of a Corey-Kim intermediate used in an oxidation reaction. And the third, where an Aryl transfer process was rapidly developed to provide multi-kilogram quantities of a desired substance. We at METTLER TOLEDO have been performing on-site lab feasibilities for over 15 years. I will describe a few anecdotes learned from these studies where ReactIR results provided a light bulb type moment. After a short summary, we will end with your questions and some discussions. We certainly welcome your questions. However, these examples were taken from the literature, and we will not be able to answer in-depth questions regarding why a particular synthetic strategy was chosen or if other experiments were attempted. During early and late process development, the organic chemist needs to gather important information from their reactions in order to understand the critical process parameters that impact the quality and robustness of their processes. This is a common theme in the pharmaceutical industry, but applies as well to other chemical industries such as ag chemicals, specialty chemicals, the polymer industry, and even towards academic endeavors. A very standard tool for the synthetic chemist is HPLC. HPLC is an outstanding analytical method for a wide variety of compounds since there is a physical separation that splits up the reaction mixture into its individual species that can then be detected with good sensitivity using either a UV detector for species with an aromatic group, say, or an ELSD, evaporative light scattering detector, for species with no UV chromophore. As with any analytical technique, HPLC is not applicable to all chemistries. There are some very basic questions that one asks during these development activities. By far the most common question is, did the reaction work? In other words, was the desired product created? This is certainly a key question during early development, where a synthetic route for medicinal chemistry may need some tweaking to make it more scale-up friendly. It is very advantageous at this early stage to have enough information, in other words, analytical data, about the reaction with which to make an informed decision regarding what the result was and what to do next. Thus, a quick and general understanding of selectivity and reactivity rates are desirable. Do I really need to wait 3 hours before raising the temperature? Also, observing any clues that may indicate potential intermediates or reaction byproducts is quite useful. Another common question is, how long did it take? Here is a case where an in situ method such as a ReactIR is really invaluable for detecting when a reaction mixture stops changing. In order to run an HPLC, a sample must be taken, prepped, and run on the column. This can be quite tedious, labor-intensive, and of course, time-consuming. Grab samples, be it for HPLC, NMR, or GCMS, provide a great snapshot at that point in time. An in situ method like ReactIR can provide a molecular video of the reaction mixture. Due to the high temporal resolution of a ReactIR, it is fairly straightforward to be able to identify an endpoint or observe if the reaction had an initiation period, or worse yet, stalled during the reaction, which potentially may lead to unwanted accumulation. A final concept that has found its way earlier and earlier in the development cycle is this process scalable? Now, this is a relative concept. It does not necessarily mean running the given process at a manufacturing scale. It may be as simple as scaling from 5 grams to 500 grams, but having to do it in 2 months. In order to scale up quickly and safely, one needs an in-depth understanding as possible within the given time frame. What are the key control parameters for this process? Do we have the proper reagents and order of addition? Can we get some basic idea of the kinetics of the reaction? Of course, the only way to answer these questions is to perform the work and have the proper analytical eyes to prove the results. As everyone knows, the picture becomes much clearer with 2 eyes versus using only 1. Employing multiple techniques such as HPLC and NMR, or HPLC and ReactIR, or even heat flow and ReactIR, provides the highest probability of gaining the correct picture of the process and doing it quickly. Now, for a brief introduction to the ReactIR and its critical sampling technology. ReactIR is a real-time, in-situ, Fourier transform mid-infrared system. The technique that we utilize in our sampling technology is attenuated total reflectance, or ATR. ATR works on the principle of Snell's law, where each reflection is actually an interrogation point of the sample matrix. At each interrogation point, the infrared radiation penetrates into the reaction matrix only on the order of approximately 2 microns. Therefore, the path length is equal to the depth of penetration times the number of reflections. Since the infrared radiation only penetrates into the matrix such a short distance, color is not a problem. For instance, the reaction can be clear or black, and bubbles and solids are not a problem for our probes. In most cases, the system will not be able to observe these solids in the sample matrix. This is quite different from a transmission-based technique where bubbles and particulates will cause interferences. In essence, the ReactIR is sampling the liquid phase of the reaction mixture. With ReactIR data, we will not plot spectra in transmittance, but rather absorbance. Why? Absorbance is proportional to concentration via Beer's law. Although A, absorptivity, and B, path length, are functions of a few variables, we generally assume them to be constant. Therefore, when a peak absorbance is plotted versus time, we are looking at a concentration versus time plot. With the specificity of the mid-infrared, a peak absorbance is associated with a particular functional group in the mixture, and we can therefore trend how the concentration of multiple species change with time in the reaction mixture. Real-time in-situ reaction monitoring, as we all know, is incredibly difficult for any analytical technique. The reason why ReactIR has been so successful, and the secret to its success, is the comp probe technology. The comp probe was introduced in 1994 and has been used successfully for over 12 years now. It is a robust technology that's been used in thousands of applications by hundreds of different companies. The design shown to the right has a zinc selenide focusing and support element that focuses the infrared radiation into the ATR sensor, which interrogates the sample. Then the zinc selenide element focuses and directs the infrared radiation back to the instrument detector. The zinc selenide element does not come in contact with the chemistry, as it is inside the metal housing of the probe. The only wetted materials are the standard alloy C276 metal housing, a thin gold seal, and the ATR sensor itself. The most widely used ATR material is by far diamond. It is an extremely hard and chemically resistant material. However, diamond does absorb in the mid-infrared between about 2250 and 1950 wave numbers. Thus, if you need to monitor in that region for components such as metal carbonyl species or isocyanates or nitriles, then we have the sensor materials of silicon and cubic zirconia available. In addition to the gold seal, we also have a Teflon seal available, and most any type of metal can be used for the probe housing. Specifications for the probe are standard temperature ranges of -80 to 200 C, with a special design that can go to 400 C. A standard pressure range of vacuum to 100 psi, a high pressure design to 1500 psi, and a special ultra high pressure design for pressures going up to 4500 psi. The comp probe design is available in 4 diameters of 6.3 millimeters, 9.5 millimeters, 16 millimeters, and 25 millimeters, and we can make those in various lengths. Contact your technology and applications consultant or TAC for help in choosing the correct probe for a particular application. When the ReactIR was first introduced, it was adopted mainly by specialist groups or engineers. Over the years, the instrument size and complexity of the software, as well as the sampling, have been greatly reduced. The ReactIR IC10 was specifically developed to appeal to the organic chemist and become a general-purpose tool in the lab. This picture shows an IC10 system in use in a chemist's laboratory hood. The system has a very small footprint, about the size of a man's shoebox. The IC10 utilizes our fiber technology integrated with our small diameter, either 6 or 9 millimeter, DICOMP probe technology. With this fiber technology, no alignment is required between reactions. Think of it as simply adding an advanced thermocouple into your reactor. Instead of recording heat, you're recording the making and breaking of chemical bonds. Another major advancement for the chemist is our ICIR software. The software is wizard-based and leads a person through the setup of an experiment. Here's a screenshot of the HPLC software. If we take a clockwise tour around the screen starting in the upper left quadrant, we have the trend view window. The trend view window is of peak profiles, which is an absorbance versus time plot of how a peak changes throughout the reaction. Also, our concert profiles can be displayed in this window. Concert is a proprietary algorithm of METTLER TOLEDO's, which takes a given region of the spectrum and a given time frame of the reaction and deconvolutes the experimental data into the components that are changing at different rates. In the HPLC software, Concert works in real time to provide profiles of components that would otherwise be difficult to observe via simple peak height profiling. Next is the event view window. It shows the date and time when a spectrum is collected and any annotations or comments added to the reaction, such as when an addition is started or when a temperature is changed. It's very important to thoroughly annotate the data set to aid in the data analysis later. The 3D view is in the bottom right-hand corner. This is the molecular video of our reaction. Here in the methanolysis of acetic anhydride, you can see the anhydride peaks up around 1800 wave numbers coming in and then disappearing, and the acid and ester bands forming around 1700 wave numbers. In the spectral view window, we can view our reaction spectra at the various points in time. Any reference spectra that we want to see, as well as the concert-generated component spectra. In this window you can see a peak identification box that gives a list of what functional groups absorb in that particular wave number region. This can be helpful to give you an idea of what a band may be. It appears when the cursor is positioned over a peak. The gray bars that you see over certain peaks are used for defining peak profiles. At these peak positions, profiles of these absorbances versus time will be generated in the trend view window. The software is surprisingly easy to use, and more importantly, easy to remember how to use and to collect a good data set. The first case study involves a Lactol activation by trifluoroacetic anhydride, which has been published by researchers at Merck Research Labs in Rahway, New Jersey. The Lactol activation shown in Scheme 1 is a step in the formation of a key lactam acetal intermediate, shown in Scheme 2, that was being used for a drug substance of interest. These reaction steps pose many challenges for process development due to the nature of the reagents and the substrates involved. Here is a case where offline sampling of the reaction mixture poses real challenges. The activated Lactol product is unstable to the HPLC prep conditions and actually reverts back to the starting material. Thus, HPLC will not be helpful for determining the extent of the reaction nor the reaction endpoint. ReactIR is an obvious candidate for cases like this where HPLC cannot or is challenging to use. Other examples of this have been discussed in previous webinars. They include reactions involving the formation of enolates or imines, anhydrides, mixed anhydrides, and ketenes. In addition, trifluoroacetic anhydride, or TFAA, is quite sensitive to ambient moisture and upon pulling a sample readily produces trifluoroacetic acid, TFA. HPLC is not rapid enough nor able to assay the content of TFAA in solution. If TFAA is overcharged, it can react with an expensive chiral alcohol, as shown in Scheme 3, to form a trifluoroacetate. Obviously, it is highly desirable to keep the cost of goods as low as possible on any project. Thus, the correct amount of TFAA to charge must be determined so as to minimize consumption of this expensive chiral alcohol. If TFAA is undercharged, the unreacted Lactol starting material can react with the activated Lactol in solution to form a DL or a mesotype dimer as shown in Scheme 4. Not only do these dimer impurities hurt yield, they can add processing time if additional cleanup is needed, say through chromatography or crystallization. Knowing the amount of TFAA is the key to following and controlling this reaction. An in situ method, such as ReactIR, is an ideal technique to evaluate for monitoring reaction behavior. A good first step towards determining ReactIR feasibility is to collect reference spectra of the reaction species. In general, it is good practice to collect reference spectra in order to determine which peaks in a ReactIR dataset belong to species X. The HPLC software prompts one to collect reference spectra with each experiment. However, one can load previously collected spectra, or collect the spectra later and reload them into the experiment file. Another very useful feature of the HPLC software is real-time solvent subtraction. Therefore, it is quite important to have reference spectra of the reaction solvent, or solvent mixture, available. Although performing automated solvent subtractions can greatly help in peak profiling, it is the best case scenario when isolated peaks from the species of interest are present. The starting Lactol material does not exhibit good solubility in acetonitrile and in the slurry at the desired reaction temperature. Since ReactIR is not sensitive to solids in solution, it will not be useful for tracking this species in situ. However, the activated Lactol shows good solubility at the reaction temperature and has unique peaks at 1679 and 1495 wave numbers. The TFAA reagent exhibits unique IR absorbances at 1875, 1320, and 663 wave numbers. Note that these peaks are away from peaks due to the acetonitrile solvent. With these peak positions, one should be able to quickly and uniquely track activated Lactol and TFAA in the reaction mixture. An initial experiment was performed using the ReactIR to record the reaction. Another good habit while collecting a ReactIR experiment is to be patient and make changes in steps if the experimental procedure allows. Not only does it greatly simplify data interpretation later on, it allows the best opportunity to learn from the IR information. A general rule of thumb is to collect several spectra before making another change. A typical data collection rate is a spectrum every minute or 2 minutes. However, data acquisition rates may be changed at any time and can be collected as fast as every 5 seconds. In this case, the ReactIR data collection began with just 88 milliliters of acetonitrile presence. Then 40 grams of Lactol, or 1 equivalent, was added to the jacketed reactor at -5 degrees C. A weak peak at 1665 wave number corresponding to the dissolved Lactol in the slurry can be observed. The reaction starts immediately upon the addition of 41.9 grams, or 1.04 equivalents, of TFAA, as evidenced by the appearance of activated Lactol peaks at 1795, 1679, and 1635, in addition to the rapid rise in the 1875 wave number peak from TFAA itself. As can be seen in the peak profile plot, this reaction is fairly rapid. The profiles level off in less than 60 minutes. Since there was excess TFAA present due to the amount that was added and the fact that the ReactIR shows a peak at 1875 wave numbers, 1 hour after the TFAA addition, an additional tenth of an equivalent of Lactol was added. Recall that no HPLC method was available for the activated Lactol since it simply reverts to the original Lactol starting material. No quantitative IR method is possible since there's no reference method for determining the absolute concentration of the activated Lactol. Like other spectroscopic techniques, ReactIR needs a reference method in order to assign a quantity to a particular spectrum. Although the unique Lactol peak absorbance is proportional to concentration, we can only use this peak profile in a qualitative manner. A final observation on this initial experiment was a fairly large exotherm of about 10 degrees with the TFAA addition time of only 5 minutes. Not only is this temperature rise a potential safety concern, it also contributes to the dimer impurity formation, which will be discussed later. Although a quantitative IR model was not possible for the activated Lactol, a quantitative model was developed for TFAA in acetonitrile. Ideally, when one develops a quantitative IR model, samples from the actual reaction mixture are submitted for reference analysis and correlated to the spectra. In this case, that was not possible with TFAA. However, since the TFAA exhibits such a strong and well-resolved band at 1875 wave numbers, a simple TFAA spiking experiment was employed. Known amounts of TFAA were spiked into acetonitrile and the IR spectra recorded. The peak area for the band at 1875 wave numbers was then subjected to a linear fit. In this case, two models were developed to predict concentrations in the range of 0 to 100 milligrams per milliliter and 60 to 350 milligrams per milliliter. These models were tested with known samples to ensure accurate predictions. The estimated limit of detection was 5 plus or minus 1.5 milligrams per milliliter. Since the quantitative prediction desired was of TFAA in actual Lactol activation experiment, it is prudent to confirm that the quantitative TFAA model developed in a binary system correctly predicted TFAA concentrations in an actual reaction mixture. To test this model, 0.9 equivalents of TFAA were added to acetonitrile at minus 5 degrees C. Then 1 equivalent of Lactol was added. Several additions of TFAA were made until excess TFAA existed in solution. Then known amounts of TFAA were spiked into the solution to check the prediction of the model. The calibration model did a good job of predicting the actual TFAA charge in the mixture. A final Lactol addition was made to react away the excess TFAA. The levels of TFAA, Lactol, and activated Lactol were known to affect dimer formation, as does temperature. The reactant addition order can be altered to observe the effect upon dimer formation. When TFAA is added rapidly in less than 20 minutes, to a Lactol slurry there is always Lactol in excess. Thus the Lactol may react with the activated Lactol to form unwanted dimers. Also, the exothermicity when this order of addition is used raises safety concerns. As discussed previously, a 5-minute addition of TFAA resulted in a 10-degree temperature rise. When the TFAA addition is changed to 1 hour, the observed temperature rise is an acceptable 2 degrees. If this order of addition is chosen, the dosing rate of TFAA is a critical process parameter since it affects batch temperature as well as dimer formation. This figure illustrates the effect of reversing the order of reagent addition. That is, Lactol is charged into a reactor containing TFAA in acetonitrile. The Lactol is added in 6 portions over the course of the reaction. When run in 3 separate batches, the maximum temperature rise observed was less than 2 degrees. HPLC analysis confirms that the dimer formation had been minimized with less than 0.15 mole % detected. 2 additions of TFAA ensure that there is a slight excess of TFAA. In addition to minimizing the exotherm, this reverse addition should help to minimize the amount of free Lactol at any time, and therefore also serve to limit the extent of dimer formation. It was discovered through separate experiments that the amount of dimer formation was dependent upon 3 items: temperature during the Lactol activation, the Lactol to TFAA charge ratio, and the post-reaction conditions. As long as the reaction temperature during the Lactol activation could be maintained between -5 and 0 degrees, the dimer levels will be less than 0.3 mole %. Therefore, effective temperature control is essential for the activation experiment at any scale. The charge ratio and the post-reaction conditions are related. Overcharging the Lactol, in other words, undercharging TFAA, above 0 degrees would favor dimer formation. For instance, when a charge of 1 to 0.5 Lactol to TFAA at -5 degrees C was used, only 0.12 mole % dimers were formed after aging for 1 hour. However, when the temperature was raised to 20 degrees C, 2.48 mole % dimers were formed when aged for 2 hours. Additional aging overnight at 20 degrees C resulted in almost 35 mole % dimer formation. On the other hand, if TFAA is overcharged slightly, the levels of dimers formed does not increase significantly with elevated temperatures. For example, with a 5 mole % overcharge of TFAA, a 3-hour aging at 30 degrees C only resulted in a 0.2 mole% dimer formation. To illustrate the complementary nature of ReactIR and HPLC to this project work, consider this quote. While the FTIR was used to determine the conditions which led to high-level dimer impurity, the amount of dimer impurity was determined by HPLC analysis. This example illustrates several advantages to augmenting traditional HPLC analysis with in situ ReactIR monitoring. Since ReactIR is a rapid, real-time technique, it's easier to identify critical process parameters and obtain some basic kinetic information early in process development. Not only can ReactIR be used as a qualitative tool, but it can also be used as a quantitative tool to monitor and control reaction processes in the development lab as well as the production plant. The topic of an upcoming ReactIR webinar is going to focus on the use of ReactIR for scale-up at the kilo lab and pilot plant scale. Just check the website for the upcoming times and dates. For this work, it was certainly the case that the two eyes of HPLC and ReactIR working together provided a clearer picture of the reaction dynamics, which led to discovering the optimal reaction parameters. The second case study involves the study of a Corey-Kim intermediate used for an oxidation process as evaluated by a team from Abbott Labs in North Chicago, Illinois. Although there are other strategies for achieving oxidation reactions, the selectivity afforded via a Corey-Kim oxidation merits its continued use for many organic syntheses. The preparation of the Corey-Kim intermediate is achieved through the reaction of N-chlorosuccinamide, NCS, with dimethyl sulfide, DMS, as shown in Scheme 1. Addition of an alcohol in combination with the tertiary amine yields the desired ketone or aldehyde. In this case, the Corey-Kim intermediate was an essential part of the desired large-scale synthesis of a ketolide antibiotic, ABT773, as shown in Scheme 2. Oxidation of the C3-hydroxyl group with Corey-Kim intermediate provided compound 3, which then undergoes a deprotection of the benzoate to yield the desired ketolide. Since the goal was to run this chemistry at a large scale, greater than 300 kilograms, the stability of a large amount of Corey-Kim intermediate was of keen concern. It has been shown in the literature that Corey-Kim intermediate can undergo decomposition via the two pathways shown in Scheme 3. Addition of heat can yield succinimide 5 and methylthiomethyl chloride 6. Upon addition of a tertiary amine, such as triethylamine, the methylthiomethyl succinimide 7A and 7B are formed. Temperature control is therefore quite important given the facts that this reaction of NCS with DMS is exothermic and the desired scale of this reaction is quite large. Maintaining active Corey-Kim intermediate is also very important, as is understanding the optimal reaction time. There would certainly be an increase in processing time once this project was scaled up. It is only logical that this increased time could lead to decomposition of the Corey-Kim intermediate. A final concern was the potential reactivity of the substrate itself with Corey-Kim intermediate, since the substrate and product have tertiary amines which could act as a base for the decomposition reaction. Thus, knowing the stability of the Corey-Kim intermediate is the key to determining appropriate reaction times and temperature. Again, in this case, the reference spectra of the species in the reaction mixture show unique IR absorbances. Although a peak was expected around 1735 wave numbers from the Corey-Kim intermediate, this peak was not well resolved from the carbonyl peaks of NCS. As is oftentimes the case with ReactIR, peaks appear in other portions of the spectrum due to the desired species of interest. In this case, peaks at 1036 and 1011 wave numbers were observed to increase with time when DMS was added to NCS and dichloromethane. These peaks are not consistent with the reference spectra of NCS, DMS, or DCM. The peak at 1011 wave numbers was used in subsequent experiments to track the Corey-Kim intermediate. The temperature stability of the Corey-Kim intermediate was investigated at a variety of temperatures, as shown in Figure 2. These experiments were carried out in a METTLER TOLEDO RC1 reaction calorimeter to ensure optimal temperature control during this exothermic reaction. The presence of Corey-Kim intermediate after 20 hours can only be observed via ReactIR in two cases where the reaction is below 0 degrees. The presence of active Corey-Kim intermediate was confirmed by adding isopropyl alcohol and triethylamine to the aged reaction mixture in performing GCMS. The expected Corey-Kim oxidation products were indeed detected. A predictive model for the concentration of Corey-Kim intermediate was desired. Therefore, a kinetic analysis of the decomposition was undertaken. Corey-Kim intermediate was synthesized at a variety of temperatures and concentrations of NCS. A model reaction was selected for the kinetic analysis since zero dilutions of Corey-Kim intermediate showed a nonlinear absorbance response via ReactIR. This reaction was the addition of benzyl alcohol to produce benzyl chloride. The residual benzyl alcohol concentration was determined via HPLC analysis. The plot in Figure 3 shows the results of this work and points to zero-order kinetics for the decomposition reaction. The two main conclusions drawn from the kinetic analysis and predictive model for maximizing the concentration of Corey-Kim intermediate were first to keep the reaction temperature as low as possible, and secondly to keep the concentration of NCS as high as possible. Of course, there were practical limits due to the scale of this reaction was to be run at. Using the predictive model and keeping in mind the capabilities of the plant, it was calculated that an NCS concentration of 0.535 molar could be used. A final detail to evaluate was the stability of the Corey-Kim intermediate to the presence of tertiary amines in the substrate. It can be reasoned that there are two competing pathways for reactivity. First, the tertiary amine can cause the decomposition of the Corey-Kim intermediate as described previously. Second, it could act as the base and cause the desired oxidation to form the desired product. Figure 4 shows that even at -8C, a tertiary amine can cause rapid decomposition of the Corey-Kim intermediate. However, when compound 2 is added to the Corey-Kim intermediate, only a slow decrease in intermediate concentration is observed, as seen in Figure 5. HPLC analysis confirms that before the addition of triethylamine, there is about 20% of the desired ketone of compound 3 present. This observation indicates that the tertiary amine of compound 2 is acting as a base for the desired reaction and not decomposing the Corey-Kim intermediate. Unfortunately, in situ ReactIR could not observe a new peak from the product directly, most likely due to overlapping ketone absorbances from compounds 2 and compounds 3. After the addition of the triethylamine, the reaction proceeds rapidly and is complete in about 1 hour, which was confirmed by HPLC. For the large-scale lot, excess triethylamine is not desirable since it consumes Corey-Kim intermediate. Range-finding experiments show that the reaction can proceed to completion in about 2 hours with one equivalent of triethylamine. Using the knowledge about the Corey-Kim intermediate gained through this study, the oxidation of compound 2 was successfully carried out at a greater than 300 kilogram scale. The third case study comes from a recent report from researchers at Eli Lilly and Company in Indianapolis, Indiana. Multi-kilogram quantities of the ether-linked diarylmethanols 1 and 2 were required for research, which is being directed towards therapies to treat acute migraine headaches. In order to supply 500 grams of material, the original synthesis was simply employed. However, in order to rapidly supply multi-kilogram amounts of material, the original scheme needed development in order to scale up. It was especially desirable to replace the chiral chromatography step in Scheme 1 with an asymmetric method to produce the chiral alcohol 7. To this end, several potential strategies emerged. The first involved asymmetric reduction of a ketone followed by an enzymatic resolution of compound 6. Another involved the asymmetric Aryl transfer from an aryl zinc species to an aromatic aldehyde. Work reported in the literature by Baum was particularly attractive since it permitted the use of aryl bromide 4 in Scheme 1 as a precursor to a known boronic acid. This reported methodology uses 130 mole% aryl boronic acid to produce the aryl zinc species. This excess was an obvious target for improvement in order to utilize this strategy on the desired scale. Also, recent solution IR work from the Paracas group had investigated the rate of phenyl transfer from diphenyl zinc relative to the phenyl transfer rates from mixtures of diphenyl zinc and diethyl zinc. It was determined that about a 28 mole% excess of phenyl groups was optimal for producing the aryl zinc species. The work in Scheme 2 was initiated to investigate whether the more favorable stoichiometry from the Paracas group could be employed with the Bohm methodology of creating the aryl zinc species. The result of the first attempt, outlined in Scheme 2, was to produce the desired diaryl methanol species 7 in about 95% yield and greater than 94% EE. Although this result is quite satisfying, there were several areas where process development would make it more applicable to the kilogram scale. Most of these desired improvements involve reducing reagent amounts and reaction volumes. In order to address these optimization goals, a multi-kilogram prep of the aryl boronic acid 13 was conducted. Instead of N-butyllithium, N-hexylithium was employed to reduce the dangers of butane release. Surprisingly, the NMR and IR characterization of the reaction product did not fit with the reported boronic acid structure of compound 13. No OH protons were present in the NMR spectrum, and no OH bands were present in the IR spectrum. It became clear that the structure of compound 13 was incorrect. The actual structure was the boroxine compound 16. This structural understanding provided by the two spectroscopic techniques was extremely important to the stoichiometry optimization required for this rapid scale-up project. Initially, the reaction between boroxine and diethyl zinc relied upon the conditions set forth in the literature, which had no method of analysis. This prep held the reaction mixture at 60 degrees C for 15 to 20 hours. In order to gain a better understanding of the reaction and to ensure that it was reproducible, a ReactIR was used to monitor the liquid phase, and a real-time gas analyzer mass spectrometer was used to interrogate the off gas. The ReactIR showed a decrease in band intensity at 1408 wave numbers upon addition of diethyl zinc at 20 degrees C, and an increase in a band at 1289 wave numbers. There was little or no further change within just a few minutes after the addition had been completed. Figure 1 also shows the corresponding mass spec data from the off gas during the addition. The rapid decay of the ethane production is consistent with the ReactIR observations. The mass spec also confirmed that the off gas is indeed ethane and not ethylene. With the diethyl zinc addition complete, the reaction mixture was then heated to 60 degrees C. The 1289 peak from the ReactIR now disappears in about 30 minutes. This mixture was shown to be active for the desired aryl transfer chemistry. Based upon these observations, the literature hold times of 15 to 20 hours at 60 degrees C after the diethyl zinc addition can be reduced to only 2 hours with no concern for irreproducibility due to an incomplete reaction to form the aryl zinc species. This time savings is obviously significant since time was of the essence on this multi-kilogram scale project. The ability to collect information from multiple analytical techniques is especially valuable for early stage projects under demanding milestones, be they delivery times or amounts or both. Indeed, this analytical information is even more critical when attempting new, unproven synthetic pathways under the same milestone pressure. In this case, the combination of techniques, NMR, ReactIR, mass spec, HPLC, provided the necessary process insights needed in order to optimize conditions relevant for multi-kilogram batches of material. I would now like to share a few anecdotes regarding some of the feasibility studies we have conducted over the years that point out how one ReactIR experiment can provide a tremendous amount of reaction insight. We all would like to believe that our chemistry is well behaved and we understand how it proceeds. On paper, that may be true. In reality, we don't know what we don't know. As was discussed earlier, One fundamental question is always, how long does this reaction take? Before every feasibility, I ask this question. Sometimes the answer is known, but oftentimes I receive a guesstimate. As you can imagine, I have observed cases where the reaction is much quicker than anticipated, such as in the Lilly example, and in a few cases where the reaction was much slower than expected. More often than not, the real reaction time that ReactIR can measure is a surprise. One customer described to me how the ReactIR was able to show that a product formation actually catalyzed the reaction. HPLC taken at time 1 showed the reaction was going, and taken at time 2 that it was done, but there was no information about how the reaction got from point 1 to point 2. The video camera nature of ReactIR data showed that the exponential rise of product to an endpoint. This reaction was performed at a small scale. One can imagine the challenges of scaling up such an autocatalyzed reaction. Another described to me how a process was exhibiting yield and byproduct issues. It was an early project, and this needed to get solved ASAP. By collecting one ReactIR experiment, they were able to see the product peak height profile plateau to a flat endpoint, at which time it started to decline. At the same time, a new peak started forming at about the same rate as the decline in the product. The quench for this reaction had not been consistent, nor applied at the correct time to ensure high yields, few byproducts, and reproducible results. Telescoping reactions has become a very desirable practice due to the efficiency of performing multiple steps in the same vessel. ReactIR makes an ideal tool to help ensure the proper amount of time is spent on each step before moving on in the synthesis. For example, a process was not demonstrating consistent yields or percent EE. In this case, similar to the Merck example, an expensive chiral alcohol was being added to a ketene species that was being generated in situ at a very low temperature. In one ReactIR experiment, we could see that the ketene was formed very rapidly but was not stable even at this low temperature. The alcohol addition was coming much too late to ensure the desired outcome and ensure that it was consistently achieved. I always encourage chemists to intentionally introduce changes while recording ReactIR data. In other words, doing little mini experiments within the main experiment. What happens if you added more base, or more catalyst, or more substrate? What would happen if the stirring stopped or the temperature was changed? ReactIR can tell you almost immediately how the system responds to the change. A final anecdote involves reagent selectivity. In this reaction, a simple halogen exchange was desired. A clear IR peak from the CBR stretch could be observed in solution. However, upon addition of a reagent, this peak did not react away as expected. In fact, a peak from another functional group in the substrate was observed to decrease after the addition. The reaction was stopped, a sample was analyzed to confirm that indeed the wrong chemical transformation was occurring. In conclusion, the case studies presented here have demonstrated how the ReactIR complements traditional HPLC sampling to provide detailed reaction understanding, which is necessary for process development and rapid scale-up. While HPLC sampling can provide superior sensitivity, the ReactIR can provide real-time concentration information regarding major reaction species. In case study 1, ReactIR was used to track the amount of trifluoroacetic anhydride in solution, which is the key to controlling and following the Lactol activation reaction. In case study 2, ReactIR was used to track the amount of chorochimine intermediate to provide stability information, which was then used to determine the proper parameters for a large-scale oxidation reaction. In case study 3, ReactIR helped provide structural understanding, which was important for stoichiometry optimization. In addition, the ReactIR was able to confirm that a batch hold time could be reduced from 15 to 20 hours down to only 2 hours. The theme weaving through all of these case studies is the use of real-time ReactIR as an orthogonal analytical technique. As any good skilled craftsman knows, you need the right tool for the job. That is why the Craftsman Toolbox is full of so many varied tools. The toolbox is brought to the worksite every day, tools at the ready. The modern synthetic lab is no different. 30 years ago, the HPLC was a nice-to-have tool, which today is found on almost every synthetic benchtop. 20 years ago, the ReactIR was introduced for in-situ reaction monitoring. Now, 4 instrument generations later, the ReactIR IC10 should be in the toolbox of every synthetic lab. As discussed in this presentation, one will be surprised what can be learned simply by putting a probe into the reactor and watching what happens.